Hi, everyone. Welcome to the first in a series of webinars, the Go with the Flow series. We're just going to wait a couple minutes here as attendees start rolling in. We'll get started in a few minutes. Hi, everyone. Welcome to the first in a series of webinars this month called the Go with the Flow webinar series. We'll be having a guest speaker to talk about their experience with the FlowVPE or VPX every Tuesday at this time for the following month. And then a final panel discussion with our CEO, Tony Hunt, where we can discuss all these applications together at once. So 5 in the series, 4 technical and 1 panelist discussion at the end. Today, we have Rashmi Bengali. She's a process development scientist from KBI Biopharma, and she's here to talk about her experience with the FlowVPE and dynamic binding capacity. So if you have any questions during the webinar, feel free to write a question to staff in the chat box. We'll receive your questions, and then at the end, we'll open it up for questions, and we can go through them after the presentation. So with that, I'll give you Rashmi and her work with dynamic binding capacity.
Thank you, Ramsey. Good morning, everyone. I would like to begin by thanking Repligen for giving this opportunity to me to be a part of this webinar series. And today I'm going to talk about the application of variable Pathlength technology in downstream process development. Before I delve deeper into this topic, I would like to give a brief introduction to KBI Biopharma. We are a contract development and manufacturing organization, and we offer services in analytical and formulation sciences, microbial and mammalian cultures, and also cell therapy. We have a wide client base ranging from small startups to big pharma companies, and we have an excellent track record of client satisfaction. Since 2015, we have been under the JSR Corporation umbrella. A little bit about how we are placed around the globe. So KBI Biopharma is headquartered in Durham, North Carolina. This is also our manufacturing facility. Then in Durham also, we have the Research Triangle Park site, which is where our process development and analytical development group operates from. And very soon, we are going to have a commercial manufacturing facility also in Durham, which is going to be dedicated to the mammalian GMP manufacturing. In the US, we also have a couple of other locations in Colorado, at Boulder and Louisville, and our cell therapy group works out of the Texas site. In the Europe, we have a couple of places where we are placed. So we have been in Leuven, Belgium for quite some time. And since Celexis became a part of JSR Corporation, we are co-located with them out of Geneva, Switzerland. So we are nicely placed in the North America and the Europe to take care of our clients in both the continents. Talking about the process development team, which I'm a part of, we have handled around over 150 programs so far. Most of our projects are related to early phase development, mostly phase one molecules, but we also have expertise in working on phase three molecules, and we routinely perform process characterization studies as well. So once we develop the process in our PD labs, we transfer it to our manufacturing facility, and we have so far completed more than 140 clinical batches successfully. On an average, KBI helps file around 13 to 17 INDs annually. And we have also had our first successful PPQ campaign recently in preparation of commercialization. So we have a very wide portfolio and expertise not only in the conventional MABs manufacturing, but also in the so-called difficult molecules. Since Celexis became a part of JSR Corporation, Celexis is a world leader in cell line technology, uh, s- cell line development technology. So since we belong to the same JSR group of companies, we have collaborated together to offer what is known as gene to GMP in 9 months. So this is a very, very aggressive timelines, and we are helping our clients reach market faster. And that is the reason why at KBI we are constantly trying to find innovations, and constantly we are on lookout for new technologies which will give us some sort of efficiency gain. So the efficiency gain can be in terms of process improvement, in terms of time saving, cost saving. uh, so that is what we are constantly on a lookout for. And similarly, when as we explore new technologies, we came across the variable path length technology, and we started exploring this technology around 3 years back. We have been constantly using the FlowVPE, which is also based on this technology, but we started using the FlowVPE around 3 years back, and it has helped us a lot. And right now, it is a part of our regular workflow. Um, so I'm going to talk about the principle on which this technology is based, and then I'm going to discuss some of the applications which we have used this for in KBI Biopharma. So as we all know, the biopharmaceutical market is growing by leaps and bounds, and the expenditure on R&D is going to increase here on. So what is important for the companies to remain competitive in this market is that we should have an edge of some sort. So getting the drug to the market sooner is always beneficial. And as I mentioned earlier, and that is the reason why we are continuously exploring new opportunities which can increase the efficiency of how we operate. Now, within the biopharmaceutical space, monoclonal antibodies occupy a very important space. And that is the reason why I'm going to focus my talk on the monoclonal antibodies purification today. So monoclonal antibody, the downstream purification is a quite significant, contributes quite significantly to the overall cost of the drug manufacturing, and we are always in a quest to sort of reduce these costs as much as possible. On the left here, you can see the platform process for monoclonal antibody purification. So we start with a clarified cell culture harvest, and we capture the protein, the target protein of interest, using a affinity chromatographic medium. So most often, these are protein A resins, which can selectively capture your protein. And then that's what follows is low pH viral inactivation, then a couple of polishing chromatography steps, viral filtration, and then the final formulation. Now, if I split up the unit operations involved in the downstream purification of MAB, we see that a major portion of that cost can be attributed to a single step, and that is the protein A chromatography step. The reason is that these resins are really expensive. So people have been trying for many years to come up with an alternative to these expensive resins, but so far we have not been able to replace the protein A resins just because they are so efficient and they can provide a pure stream greater than 90% purity in a single step. So now that we know that we have to use a protein A resin in our MAP process, we try to make sure that we are trying to keep the cost of this step less so that we can bring down the overall cost of our downstream process. So what is important when we develop the affinity chromatography step, which is also what I call as the primary recovery or the primary capture step? So what is important during the development of this step is that we have to make sure that we are utilizing the resin to its maximum capacity. So for the entire downstream process to be economically very sound, it is necessary that we are making sure that we are not letting any capacity of the resin go waste. Now, what tells us what is the capacity of the resin? It is the dynamic binding capacity. So to make sure that the resin is used to its max, we have to first determine the DBC. And that is the reason why dynamic binding capacity experiments are the first experiments which we perform when we start process development for any molecule. So this is a very important step and helps us in resin selection. So once we have selected our resin based on the DBC, we can proceed further on the optimization path. So higher DBC means we are able to process more protein using a less volume of resin. That means we are using the resin to its max capacity. And what it does is lower the overall cost of your drug. So from here, we can see that the accurate determination of DBC for the protein A resin is very important to have an efficient process. The formal definition of DBC is that it is the maximum amount of target protein that can be loaded onto a chromatographic resin without losing product. So higher DBC means we can process more protein with less volume of resin. Now, this makes a lot of difference when we are talking about scaling up the process. So when we scale it up to manufacturing, we are talking about hundreds of liters of the protein A resin, and higher DBC gives us an edge there. It tries to bring down the overall cost. So now, since this is such an important aspect of a downstream process, now how do we determine DBC? So DBC is determined under actual experimental conditions. So what is done is that your feed stream containing your target protein is passed through the column and passed to the column, and your protein effluent coming out of the column is monitored. So initially, as the protein goes into the column, it is bound completely by the resin, and we see zero protein in the outlet of the column. But as the resin starts getting saturated, you can see some of the protein breaking through, and it can now be detected in the column outlet. So slowly, the concentration of the protein in your outlet streams goes on increasing till a point where we see that the outlet concentration is the same as the inlet concentration. Now, if you plot a curve of the concentration as a function of time, you see that you get a sigmoidal curve, and this curve is known as the breakthrough curve. The breakthrough curve is usually plotted by fractionating the flow-through fractions which are coming out of the column, and then these fractions are analyzed for the target protein concentration. And once the breakthrough curve has been plotted, the 10% of that breakthrough curve is what is typically designated as the dynamic binding capacity. So theoretically, this is the amount of protein which can be loaded onto a column without any wastage. But companies want to be a little safe. So they add a factor of safety into this. So many companies use 80% of the 10% DBC value as their maximum loading capacity. We at KBI also use 80% of the 10% value. If you want maximum utilization, you can also go for 90 to 95% of these value. So basically, based on what these 10% DBC value is, we select what resin we want to use for further process optimization. Now, the DBC is determined for all the chromatographic steps. So it is determined for the primary capture step as well as all the subsequent polishing steps. But there is a big difference. So for the polishing steps, let's say I'm determining the DBC for a cation exchanger or an anion exchanger. I'm dealing with a relatively pure feed stream, and the protein in this feed stream can be directly quantified using UV absorbance at 280 nanometer. Since the protein is relatively pure, you can get real-time concentrations based on only these values. So DBC determination for the polishing chromatography steps is not, let's say, as challenging as it is for the proteinase step. So what makes it challenging to determine the DBC for the proteinase step? The feed which we use for the proteinase step is clarified cell culture harvest. And this is the most impure stream in the entire downstream process. So it contains a lot of other impurities. It contains host cell proteins and DNA, and it keeps on adding to the absorbance at A280. The actual target protein is present in really small quantities in the cell culture harvest. So as a result, we cannot monitor the target protein concentration in the effluent during the DBC determination for a Pro-A resin. Now, you would argue that we can use a pure protein for DBC determination of protein A resin. And yes, we can do that. And academic settings often use pure proteins for determination of DBC on Pro-A resin. But then the industry standard is to use the clarified harvest. Also, because of the presence of so many other impurities in the clarified harvest, there is definitely going to be a difference between the DBC obtained using pure protein and DBC obtained using the cell culture harvest. So it is always better to use the cell culture harvest as your feed stream for DBC determination. So now that we know we cannot use the A280 methods for the concentration determination, what we have to do is we have to fractionate the column effluent, and we have to submit these fractions to for analytical testing, where they determine the concentration in the fractions using some other method. So most often it is a Pro-A HPLC titer method. And then when we get back these results, we plot our breakthrough curve. So what happens here is that there is a lot of resources involved in just analyzing those many fractions. Also, it takes quite a lot of time. Plus, it is very operator intensive. So there are a series of dilutions to be made, and all these things can basically introduce error into your calculations. And the thing which we are most concerned about is that it increases the overall process development timeline. There are some other disadvantages also of the traditional method of fraction collection, and that is that the breakthrough curve is dependent to a large extent on the frequency and the volumes of the fractions you collect. And sometimes we see that we do not get a nice smooth curve. The curve is very discrete. I'm going to show an example in the very next slide. But these are some of the disadvantages of using the traditional methods. So here you can see this is a DBC. On the left, you can see that this is a DBC experiment which we had performed. We typically use a FPLC system, an ACTA Avent, for performing our experiments. And this equipment is equipped with a inline UV detector, which gives us the UV trace of the column effluent. Now, these detectors are fixed path length detector. So they can only measure concentration up to a certain limit, above which the detector gets saturated because of the fixed path length. So when we start flowing the feed stream through the protein A column, initially what we see is all the protein impurities coming out, and we get a baseline absorbance corresponding to the host cell proteins and DNA and all the other impurities. Now, this absorbance is usually so high that it saturates the UV detector. And then when your protein starts breaking through, you can see an increase in the UV absorbance, but then there is no way we will be able to calculate 10% of breakthrough accurately based on this UV spectra. So also, I think one point of importance here is that the feed titer matters a lot. So if you have a good titer, like let's say 2 to 3 grams per liter, you can see a nice distinct increase in the UV absorbance. But when we are talking about low titers, let's say less than 1 gram per liter, this you will not even probably see this type of increase in the UV absorbance. So this is one of the problems which is associated with the inline UV detectors, which are fixed path length. So what we do is we collect all these fractions and get them analyzed offline. And then based on the concentration we get, we plot a breakthrough curve. Now you can see here that the fractions were collected and the points jumped from 0% breakthrough to 13% breakthrough immediately. In a single fraction, the concentration changed from 0 to 13%. So basically, we did not get a point at 10% breakthrough. We obviously plotted our breakthrough curve and we interpolated our data to get the 10% breakthrough. But I will not be confident in saying that this is the precise value. So you can see that using traditional methods, there are a number of disadvantages which we have faced. In addition to the time and the resources used, we are also sometimes not able to predict the DBC accurately. So the answer to that is the use of variable path length technology, which basically removes the disadvantages associated with the fixed path length inline UV detectors. You can see on the left that this is a traditional UV detector which has a fixed path length. And as the absorbance of your effluent increases, what happens is, as the concentration of your effluent increases, you see that the absorbance also increases. And since the path length is constant, the absorbance quickly falls out over the linear range of your Beer-Lambert's law. Now, since it is no longer in the linear range of the Beer-Lambert's law, we are not able to get correct concentration for that particular sample unless we dilute it. But when you look on the right side, the variable path length technology basically has a flow cell through which your effluent is continuously flowing. And then this is a fibret through which the light is transmitted. The fibret can move up and down in the flow cell, and this is how the path length is adjusted. So based on your concentration, the path length is adjusted in such a way that the absorbance we get is always in the linear range of the Beer-Lambert's law. So what happens here is that we can get real-time absorbance of whatever stream is passing through this particular flow cell. There is no need for offline concentration determination. So that is the single most advantage point of this particular technology. So let us see. This is an instrument called as FlowVPE, which is based on the variable Pathlength technology I just discussed. The fibrate can auto-adjust its path length from 0.005 millimeter to 8 millimeter, so that the absorbance always remains in the linear range of the Beer-Lambert's law. It uses a slope-based technique, and it can acquire multiple data points within short intervals of time. And if we know the extinction coefficient of a target protein, we can get real-time concentration values. Now, how have we applied this for DBC determination? So, since our major problem was the oversaturation of the fixed path length detection, we have implemented this with our FPLC systems in such a way that we get real-time concentration of our effluents, and we plot our breakthrough curve immediately after our experiment is done. So this is how we have applied the FlowVPE system for the DBC determination of the protein A step. To date, we have applied this technology for greater than 20 molecules, and it is increasing every single day. So how this is done is that the outlet of the column is routed through the flow cell of the FlowVPE, and then the outlet of the FlowVPE is routed back to the FPLC system. This is the fibrate, which goes up and down and continuously keeps on changing the path length. And the system, the FlowVPE system itself, is connected to a dedicated software which continuously acquires the data and which can provide you the breakthrough curve real time. So this is a glance of what the data acquisition using FlowVPE looks like. So while the experiment is running, there are a number of parameters which are continuously monitored. The first one is the absorbance versus path length. So since the path length is continuously changing, we can see how it is continuously tracked. Then there is concentration versus time data, which is what we use for DBC determination. And then there is also a R square versus time data. R square is basically a factor which tells us how accurate the readings are and how well your system is performing. And then there is slope versus time. So this is how the data is acquired. And when we end the run, we get the data in a tabular form. And we use the minutes versus concentration data to plot our breakthrough curve, which I'm showing in the next slide. So here, this is the data which we acquired real time using the FlowVPE. So on the x-axis, you have your time. And since we knew the extinction coefficient of our target protein, that was also inputted into the software. As a result, we were directly able to get the concentration in real time. You can see that this straight line actually is the baseline concentration, and it corresponds to all the impurities present in the feed stream. So this is the concentration of everything in the harvest except your target protein. And once your target protein starts breaking through, the concentration starts increasing above the baseline concentration. And you just need to subtract any of these values. You just need to subtract the baseline value from any of these values to get the actual concentration of your target protein in the effluent stream. So once we have this data, we incorporate the harvest titer, the column volume, and the flow rate to plot the breakthrough curve, which is something like this. So you can see that it's a nice and smooth breakthrough curve with multiple data points for every time point, and we can accurately predict the 10% DBC using the breakthrough curve. And these are generated almost real time. So when we started exploring this system, in KBI Biopharma, the first thing which we wanted to do was that we wanted to show that this system performs very well and comparable to the traditional methods. So the first set of experiments which we performed were to make sure that the data from both the methods compared. So we did those experiments, and then we also wanted to make sure that the system works well for all the concentration ranges which we see for our harvest titers. So there were concerns about low titer harvest, since generally we tend to have more noise for low harvest titers. So we tried it with low harvest titers, intermediate titers, and high titers to see if it is comparable. So that was the next set of experiments which we performed. Then we applied the FlowVPE for different types of molecules which we work on. So we applied it for monoclonal antibodies, fusion proteins, bispecific monoclonal antibodies to see that they are giving us comparable results with the traditional methods. And once we had established the comparability, we moved on to the resin screening and process optimization using the FlowVPE. We have used it for resin comparison, we have used it for flow rate comparisons, and we have also used it for resin lot to lot variability assessment. And we are also planning a resin reuse study using this technology in future. So let's look at how the data from the traditional and the FlowVPE compare. So on the left, you can see a graph where the traditional offline titer method was used to plot the curve with the blue points. So it's a discrete curve. And then the brown points are the data which we got from the FlowVPE. So it's a nice smooth curve, and you can see that they are very close to each other. So if we determine the DBC based on this, they are going to return back more or less the same value. So we know now that it is comparable. So we did that for a number of different molecules, and some of that data is present in this table. Uh, as I mentioned earlier, we were concerned about low titer feed streams. So we used a FC fusion protein having a titer, clarified harvest titer of 0.6, and we determined the DBC using both the methods. And we saw that the variability was around 5%. We repeated these experiments for intermediate titers and a high titer, and we saw that the variability was always less than 10%. And this variability is acceptable to us for multiple reasons. The first reason is that we have a factor of safety built in when we decide our maximum binding capacity, maximum loading capacity. So we have a factor of safety built in, and that is a reason why a little bit of variation is not going to make or break our process. And the second reason, I think, which is more important, is that these results can be obtained very fast. So we are ready to take this much of variability because we can save considerable amount of time. So you can see that it worked very well for FC fusion protein. It worked well for a bispecific antibody, and it worked well for monoclonal antibodies. So we have a lot of data, but this is like just a part of the data. But all of this data proves that the traditional method and the FlowVPE method perform comparably, and there is no reason actually why we cannot extend this to other recombinant proteins as well. So once we have established that we see comparable results using both the methods, we adopted this technology into our workflow, and we have routinely used this for our resin screening and process optimization studies. In the first graph, you can see that we have compared two resins using the FlowVPE, and the breakthrough curves are literally overlapping with each other. So this tells us that the DBC, the maximum binding capacity of both the resins, is the same. So But still, a point worth mentioning here is that, it does not make resin selection difficult if you look at the cost of the resins. So there is a high probability that though both the resins gave us the same DBC, one of the resins is cheaper than the other one. So here, the selection is driven by the cost of the resin. So that is one aspect for resin selection, definitely. Then in the second graph, you can see that there is a difference in the DBC obtained for both the resins. And here, we would definitely go for a resin which is giving us a higher binding capacity. Now, this table combines multiple resin screening experiments we performed for different molecules. We typically screen 2 resins. We have also screened 3 resins for some molecules. So this gives us a nice breakthrough curve comparison and helps us in resin selection. So one point I wanted to highlight was, when you look at these curves, there is so much of difference between the the DBC of these 2 resins, that the resin decision is so easy here, and will definitely lead to a more economical process if we select the blue resin. And the best part is that FlowVPE helps us generate these curves very fast. So on an average, we spend around 8 hours per experiment. That means that we can screen 3 resins in a single day. And since there is no analytical data involved here, we can immediately select our resin and move on to process development or process optimization from the very next day. So earlier, we had to literally wait for a week to 3 weeks to get back the data showing which resin performs better in terms of DBC. And once the resin selection was done, only then we could proceed to process optimization. So here, that decision making has been reduced to a single day of experimentation. So some other case studies which I wanted to show was, the first was obviously same as the last one, which is the resin selection. Here, the selection will be driven by the cost of the resins, since most of them are showing the same capacity. Then the second graph shows how the resins perform under different flow rates. So we know that, we always want to operate at a higher flow rate just because it gives us a higher throughput, and it helps to reduce the operating time. But then at the same time, if we increase the flow rate, the DBC is reduced. So we wanted to make sure that the residence time is not affecting your DBC to such an extent that it will cause us protein loss. So we determined the DBC at different residence time to select the most optimum operating flow rate. So that is another application. And then here you can see the comparison between 2 resin lots. The breakthrough curves are so nicely overlapping over each other that we can say that these 2 resin lots perform the same. So it just provides us a good tool to uh, to have breakthrough comparison between different resins, different lots, and at different flow rates. One potential application of this instrument we are looking at is the resin reuse study. um, so as I mentioned, the affinity resins are very expensive resins, and biopharma companies want to reuse these resins as much as possible to reduce the cost associated with it. So we can use this for resin reuse, like, after every 10 cycle of use, we compare the DBC, and if we get a nice overlapping curve, you can say that the resin is still good to go. It hasn't lost any performance. So there are many applications which we have seen and which we are also exploring. So this is a direct comparison between the traditional and the FlowVPE method. So this was the very first graph I showed in my earlier slides, that the blue points are the traditional pro-A titer method breakthrough curve, and the concentration jumped from 0 to 13% immediately without a 10% breakthrough point. And then you can see the FlowVPE data here, which is a nice smooth curve. So I can definitely determine my DBC more accurately using FlowVPE than I can using the traditional methods. So theoretically, if you have infinite fractions pulled using your traditional method, your breakthrough curve should overlap with the breakthrough curve which we obtain through FlowVPE. But the problem here is that the number of the number of fractions which have to be analyzed increase exponentially, and it will take that much of time for data turnaround. So that is a definite disadvantage of the traditional methods. Now, and this is a definite advantage of FlowVPE, that we have greater precision in predicting our dynamic binding capacity. Now, this is a slide I put together to assess the time saving and the cost savings which we have realized by the use of this technology. So let us look at the time saving first. So the experimental setup for FlowVPE is very easy, whereas for the traditional methods, it takes a long time to fill in your fraction collector. So since we are collecting the flow through fractions, we have to literally unscrew each tube and put it in the fraction collector. It just adds on to the time. The actual experiment time is the same, but due to the sampling, there is added time in the traditional methods. The most important factor here is the analytical testing time. So that is the highest contributing factor to the time increase. 20 hours is roughly the time which is used to run the samples, but in practical, in actual, we have seen the data turnaround takes around 1 to 3 weeks. So that is where the impact is the most. Whereas for FlowVPE, we get the data real time and we are not waiting 2 weeks for the resin decision. So we can just move ahead and put that time to further process development and optimization. Looking at the cost impact, the FlowVPE does have a capital cost of 90,000, and the HPLC instrument also does, but I haven't included it here just because the HPLC cannot be considered as a dedicated equipment for DBC determination. But other than that, the FlowVPE has very few consumables cost. It does not use HPLC columns, it does not need buffers, and most important, it does not need additional labor. So we don't need analysts to dilute the sample, run the sample. So all that cost is eliminated. And the number of analytical samples, like if you want to screen three different resins with around, let's say, 30 fractions per resin to get a nice smooth curve, we are looking at a lot of samples. And the majority cost and time is associated with the analytical turnaround time. So all that is eliminated using the FlowVPE. So that is a very that's a lot of efficiency we have gained in the past couple of years. And then we calculated that for a CDMO like KBI Biopharma, we handle around 10 to 15 projects a year, and we are able to break even in terms of the cost in around 6 to 7 projects. So within less than a year, we are able to get back however much we have invested in this instrument. So at summarizing the advantages which we get from using the technology is that we have found an alternative for DBC determinations, which is very, very rapid, and it has completely eliminated the dependency on offline methods. And because there is no analytical assessment here, the experiment is almost operator independent. So there are less chances of errors here. Then we get a better DBC curve. We can predict our DBC more accurately, and it has helped us, screening a number of resins, and the variability is very less. So we do get comparable results there. Just in the last slide, I discussed the time and cost savings. So because there is no analytical support dependency here, we save all the time associated with that, and we can start our optimization faster. And what happens is that this reduces the overall development timelines. And then FlowVPE can be used as an important process analytical technology tool, a PAT tool. We haven't really tried this out, but it can be used in the manufacturing setting, and it can definitely be used for operations like UFDF. So because this equipment is so easy to operate, and because it provides such rapid generation of results, we are also exploring future applications for this technology. And to conclude, we have used variable path-length technology successfully for rapid determination of DBC for protein A resins. It has helped us accelerate the process development timeline. And at the same time, we have also realized significant cost saving. And we are exploring further applications to harness the power of this technology for future uses. I would like to thank the KBI upstream, downstream, and analytical development teams. Um, and this is our downstream PD group at KBI. Thank you so much for your patience. Thank you.
Thanks, Rashmi. Really great presentation. Thanks. It's very informative. We do have a number of questions for you. So why don't we just get started with those? I think there's a bit of a theme in the questioning, but I'll start with a couple of them. Do you have experience with monoliths? And if so, how do you determine the DBC in this case?
We haven't tried it with monoliths. So far, we have always been working with the conventional resins.
And then, so the next question is, and there's two that are kind of along the same line, so I'll combine them. One says, couldn't you use a smaller UV path length? And actually, specifically, have you made a comparison with a smaller UV cell, like a 0.5 millimeter cell?
No. So the ACTA FPLCs which we use, their UV detector has a fixed path length of 0.2 centimeter. So we can, of course, increase this path lengths in some other detectors, but I would still think that it would be saturated given the high concentrations of the proteins in the harvest. So even if we increase the path length, we are still going to hit saturation at some point. Does that answer the question?
Yeah, I think it does. So then the next is there any correlation determined between Pro-A HPLC concentration and FlowVPE in the spent media flow through? And I believe you showed some of this data, but I'm not sure.
Yeah, so just give me a...
That one came early on in the presentation, so maybe you hadn't gone over it at the time.
So basically all this, yeah. So here is the comparison between the Pro-A HPLC titer method and the FlowVPE. So these points which you see are actually determined using the offline HPLC titer. And even this particular one, the blue points correspond to the offline titer method. So ideally, they are supposed to be the same. The effluent concentration measured using offline titer and FlowVPE should be the same.
Okay. So then a couple more utilitarian questions here. How long can a single fibrette be used? And I know I can answer that, but I'd like to hear your thoughts on that.
Yeah, I mean, if it is handled correctly, it can be used for years and years together. So if it is handled, there is no breakable part as such in the fibrette, and if the instrument is handled correctly, we can use it for multiple runs. Like we have used a fibrette for a couple of years without any problem. And if it helps, we have a preventive maintenance every year on this equipment, and we do get vibrate with every PM. So it works fine.
And then what kind of maintenance do you do between cycles?
We just make sure that we are cleaning the flow cell properly, because there is a chance that the spent medium can go into the fibrate and can sort of precipitate in the flow cell. So we just make sure that we are flushing it out with a lot of water, and we make sure that if needed, we are flushing it out with a little, with 0.5 hydroxide solution also. And then before every use, we load the cell again, and then we check transmission. And if the transmission passes, it is good to go. So that is really all the maintenance we do, that we have to make sure that we are storing the flow cell clean.
Great. So I see that that's kind of the list of questions. I'd like to ask my own question.
Mhm.
Could you foresee a situation in the future where this type of method is used in real time during manufacturing, or maybe in a batch mode, or maybe even in a continuous mode where you could put this instrument in line and actually do real time column loading?
Yes, yes. I definitely foresee the use for that type of use for this instrument. And I also understand that these instruments are made GMP friendly, so we can definitely monitor the column effluent concentration and take important decisions based on that.
Great. If there's no more questions, I'll wait one more second here to get more questions. If there's no more, then we'll close the call. But I just want to give it a reminder that this time, every Tuesday, we'll have a new speaker on a new application of the FlowVPE or VPX technology. And please come for all of them and listen in, ask your questions, and hopefully hear about this technology and its use in industry. One popped in here. Which decisions you mean for production? So I
Um,
think talking, so we said, potentially doing real-time column loading. So I think he's asking what decisions within production do you see where we could use the FlowVPE?
so, I mean, I don't have much experience with continuous chromatography, but we can definitely use it for real-time decisions when we are doing UFDF at large scale. And I mean, if for a chromatographic operation, like if you want to stop collection when a particular concentration has reached, or if you want to start collection, or if you want to switch columns, if a particular concentration has reached, the Flow VPE can provide that to you real time to enable like those type of decisions. Like, if you are using two columns in tandem and you want a decision on when to switch based on the effluent concentration, then Flow VPE can help in there definitely.
Yeah, and I can add a little color to that for sure. Folks out in the industry, they're using the FlowVPE for exactly what you mentioned. Even just looking at the elution peak after a chromatography process to quantify the the loading mass and the elution mass, sorry. And then also for UFDF, like you mentioned, and then also for the dilution step in the drug product suite, where they would use it to make a decision when to stop the dilution when they've reached the final concentration. And then we have one other question here. What if we used A300 instead of 280 and a small path length like 1 millimeter, then correlate the protein concentration between 280 and 300 for pure protein?
So I think when we are talking about the Flow VPE, the application is more for an impure stream, like clarified harvest. Uh, like I mentioned earlier in my presentation that determining DBC for pure protein stream is really not a problem, because we can get accurate UV readings of the effluent. It is just that when we are using the clarified harvest, because there are so many impurities in the form of host cell proteins and DNA, that we are not able to get correct UV absorbance for the target protein. But in the pure streams, that is really not a problem.
Yeah, and I think I understand it pretty clearly that if you can use your real cell culture harvest for this, you will get a more accurate representation of the DBC than if you just used a pure protein to load your column, right? Am I catching that
Yeah,
right?
so as I mentioned earlier, that yes, you can argue that we can use pure protein, but then the presence of the other impurities are also going to impact how your protein binds to the column or binds to the resin. So I think to get the accurate representation, it is important that we use the most representative stream instead of using a pure protein stream, because it is definitely going to have an impact on your GBC. I hope
So
that answers.
I,
Yeah.
I have another question that's just popped in. It is this data already published? As someone on the call would like to cite some of the work that they're doing similar experiments, they'd love to cite your paper. So is this published or being published?
It is not published, but we are in the process of trying to get it published.
Okay, so I guess stay tuned on that one, and I'm sure you could reach out to us at Repligen or Rashmi to get the publication when it comes out.
Yeah, and it does have a little more data than what it was shown here, and it does touch on many more nuances of this technology.
So I guess with that, we are actually right at time. And if there's no more questions, I appreciate everyone's attendance. Thanks, Rashmi, for giving your presentation. It was really great. And I hope more can come on the next series next Tuesday. And with that, I'm going to close the call. Again, you can always reach out to us, your local rep or applications person or Rashmi directly for more information on any of this stuff. Thank you very much.
Thank you.